Here, we learn about the process of DNA replication. First of all, what is DNA? DNA is the molecule composed of two polynucleotide chains that coil around each other to form a double helix carrying genetic instructions for the development, functioning, and growth and reproduction for all known organisms and many viruses. The two strands of DNA are known as polynucleotides as they are composed of simpler monomeric units called nucleotides. Each nucleotide is composed of one of four nitrogen-containing nucleobases: cytosine, guanine, adenine, thymine. The nucleotides are joined to each another, a chain, by a covalent bonds known as phosphodiester bonds. In molecular biology, DNA replication is the process of producing two identical replicas of DNA from one original DNA molecule. The first step is to separate two strands, which done by an enzyme called as DNA helicase, and results in the formation of replication fork. The single-stranded binding protein stabilizes these single-stranded DNA. Then the enzyme primase starts the process. This enzyme makes a small piece of RNA called as primer. This marks the starting point of the construction of new strands of DNA. The enzyme called as DNA polymerase binds the primer and makes the new strand of DNA. Polymerase can only add DNA bases in one direction as 5' end to 3' end. One of the new strand of DNA, the leading strand, which made continuously, DNA polymerase adding one by one continuously in 5' end to 3' direction. The another strand is lagging strand cannot be made continuous because it runs in opposite direction. The DNA polymerase can afford only make these strands in series of small chunks called as Okazaki fragments. Each fragment is started with RNA primase and DNA polymerase add the bases in 5' to 3' direction. Once a DNA has made, the enzyme exonuclease removes the RNA primase. Then the DNA polymerase then fill the gap between these two Okazaki fragments. Finally, the DNA ligase seals the DNA fragments in both strands to form continuous double-strand DNA molecule. Thus, the DNA replication is semi-conservative type. After replication, one strand be old and one strand be a newly synthesized strand. Thanking you very much.